What happens to drugs after expiry? Do they remain physically stable? Does their potency decrease? And more importantly, will they still be efficacious after expiry? These are important questions many of us have asked ourselves during the course of our professional career. In our study, we have tried to explore and find answers to these questions.
As per regulations, all pharmaceutical manufacturers are required to place expiry dates on their products before those products are marketed. Drug products typically have an expiry date that extends from 1 to 5 years. Now, how is expiry date determined? Most of the times, the expiry date is determined by what are called as accelerated stability tests, which monitor the product over a period of time under varying storage conditions of temperature and humidity. The degradation pattern and the estimation of the degradation rate are then modeled and utilized as a part of the process for assessing shelf life. The input thus derived is used to extrapolate and arrive at the expiry date of the product. Legally speaking, the expiry date of a drug is the final day that the manufacturer guarantees the full potency and safety of a drug. That means if the manufacturer has given the expiry date of a drug or product as November 2022, that means till 30th November 2022, the manufacturer guarantees that the drug is going to be safe and potent. But what happens after November 2022? No one knows. This is because the regulations do not require them to determine how long medications remain potent after that. They just have to give a date till which they can guarantee potency and safety. After that, they need not test. This allows the manufacturers to arbitrarily establish expiry dates without determining the actual long-term drug potency and stability. There are a few anecdotal studies in the literature that indicate that the actual shelf life of the drug may be much longer than is suggested by the expiry date. This aspect holds tremendous significance not only for the patients but also the healthcare professionals and regulatory agencies, and can lead to a paradigm shift in the way drugs are used. Hence, we planned this study. The aim was to try and answer the question: what happens to drug after expiry? Do they retain their chemical potency, physical stability, and efficacy even after their expiry? We studied commonly used analgesic agents from the NSAID group. There were three oral preparations, namely tablet ibuprofen 200 mg, tablet diclofenac 50 mg, and tablet piroxicam 20 mg. And there were two injectable preparations, injection diclofenac 25 mg per ml and injection piroxicam 20 mg per ml. All these preparations were procured within the last 3 to 4 months of their shelf life. The drug samples belong to one single batch to ensure uniformity, and they were stored as per the manufacturer's recommended conditions and conforming to Indian Pharmacopoeia guidelines. During the course of our study, we investigated the following parameters: chemical potency, physical stability, and efficacy. All these parameters were studied before the drug got expired, then immediately after the expiry date, and then every 6 months till 2 years post the expiry date. The study drugs were subjected to analytical assays to estimate the active ingredient in the pharmaceutical preparation. The estimation of the chemical concentration was performed as per IP by NABL certified laboratory. As per the study protocol, the chemical potency was tested before expiry, then immediately after the expiry, followed by every 6 months beyond the expiry date for the next 2 years. The chemical concentration and potency was considered acceptable if the active ingredient was within the acceptable ranges as per the Indian Pharmacopoeia. All oral preparations have to first get disintegrated and dissolved before they can be absorbed. Hence, these important parameters were also measured using validated equipment. Analgesic efficacy was tested using animal models of pain, namely the radiant heat tail flick method and the acetic acid induced writhing method, both of which are well validated models for testing of analgesic efficacy. 6 albino mice of either sex were used to estimate the analgesic efficacy of these drugs at each point in time.
The image shows the setup for the radiant heat tail flick model. In this, the tail of the mice is kept just over a nichrome wire, but not in direct contact. Then, current is passed through the wire, and as the wire gets heated, the radiant heat reaches the tail of the mice. When the mice starts feeling the pain, it flicks its tail, and this time, which is usually in seconds, is noted. An increase in this time by a test drug is taken as the measure of its analgesic efficacy.
We can see here the setup for the acetic acid induced writhing model. Acetic acid, when injected intraperitoneally in the mice, stimulates an inflammatory response, and then the abdominal muscles of the mice go into what are called as writhes. For an analgesic agent, a reduction in the frequency and number of these writhes is a measure of its analgesic action. The table shows the results of the estimation of the active ingredient of the drug formulations. As can be seen, for all the drug formulations, at every point in the study, that is before expiry, at expiry, and every 6 months after expiry till 2 years post expiry, the active ingredient remained within the acceptable limits, proving that the drugs remained chemically potent even up to 2 years post their expiry date. Furthermore, all the oral formulations had their disintegration times well within the reference range as per IP, proving that they remained physically stable. The table shows the results of the analgesic efficacy with the radiant heat tail flick model. As can be seen, for all the drug formulations, there was no statistical difference in the analgesic efficacy at different points in the study, right from before expiry till a period of 2 years post the expiry date. Similar was the case with the acetic acid induced writhing model. Even here, for all the drug formulations, there was no statistical difference in the protection provided by these drugs at any point during the study, thereby proving that the drug formulations remained effective in relieving the pain even up to 2 years post their expiry date. The results of our study indicate that the NSAIDs have retained their chemical potency, physical stability, and efficacy even up to 2 years after expiry. This suggests that the determination of expiry date as done currently may not be the best reflection of the actual shelf life of the drugs. This, however, should not be construed as a recommendation to use drugs beyond their expiry date. However, based on our results, we propose that the process of determining the expiry date be revisited to reflect more accurately the real shelf life of a drug. We also recommend establishment of a formal extension program in which an independent organization regularly tests the drugs at a designated point of time before the expiry date and then consider extending the shelf life depending on the results of these testing programs. Thank you very much.